Hello, everybody. My name is Sahar El-Etr. I'm a biomedical scientist and a microbiologist at Lawrence Livermore National Laboratory. Today, I'm going to talk to you about a new technology where we're going to use amoeba cysts as natural containers for the transport and storage of pathogens. We got tasked with this by the government because they needed better methods for the transport of pathogens from the field to the laboratory at room temperatures without losing the viability of the actual pathogen of interest. Even though most people just get the DNA and that's fine, sometimes you actually need the pathogen itself to do additional characterization. Now, existing methods for transport in the field are basically a modification of a swab with a transport medium associated. And usually, if you're thinking of storage containers are cryogenic vials that are typically frozen at -80 degrees. Sample cleanup and enrichment now occurs only in pathogen-specific transport or culture media. And these are virtually good for up to 72 hours only in the field, and then you have to get them into the laboratory. And even then, you deal with a lot of loss of actual mobile elements in the pathogen. So we're basically proposing to use nature's own natural containers for this. Environmental amoeba, which are present everywhere in the environment, in tap water, in the soil, everywhere, typically exists as trophozoites, which is what you can see in your left. And this is what the active feeding form are. And when you stress them or you deprive them from food or you expose them to changing temperatures or predations, they go ahead and make these cysts, or they encyst. And basically what that is a very thick polysaccharide layer resembling cellulose that is very, very tough and extremely resistant to changes in the environment. So this technology basically takes advantage of the fact that many pathogens are known to survive in these amoeba cysts for many, many years and to emerge when the conditions are better and the amoeba actually exist. So we basically decided to make use of Mother Nature, and we have developed the first living system for the transport and storage of pathogens. So the idea here is if you are in the field or in a situation where you need to take a patient sample, and in a lot of cases you're away from a proper microbiology lab that can do that, the actual assessment for you rapidly, you will have this device that will contain on the bottom amoeba in a buffer that simply does not provide additional nutrients for the amoeba, so the amoeba are encysted. And in the top, you have your rich buffer and a swab system. You take your sample using the swab, and what happened is once you inoculate this device, your pathogen of interest is represented here as a red bacteria, while your skin or environmental contaminant is represented in green. And once you break this membrane in the middle of the two compartments, the rich medium in the top makes contact with the amoeba, and so the cysts will actually exist and make the active feeding trophozoites, which will feed on both your pathogen of interest as well as your contaminant. Because amoeba naturally eat bacteria in the environment, they're going to go ahead and eat any contaminant while only your pathogen of interest will survive. It will go ahead and replicate, and then eventually the amoeba, the trophozoite, will run out of food and it will go ahead and encyst. Once you reach that stage, you basically, this container containing sample can sit there at ambient temperatures for any length of time you want till you have the time to transport it into the laboratory. And then once you get to the laboratory, you basically reverse the process. You go ahead and add rich medium, and the amoeba will naturally spit out whatever pathogen is in it. And then you go ahead and you proceed normally with pathogen ID and recovery. The advantages of this system is, as I mentioned, that it can basically exist at any ambient temperature. We have demonstrated the ability of this system to basically sit in the environment for anything from 1 to 6 weeks at temperatures ranging from 4 degrees to 42 degrees with various humidity conditions with no loss of pathogen viability. More importantly, we can detect as few as 10 bacterial colony forming units, which are really 10 individual bacteria. The system can detect gram-negative bacteria, for example, Acinetobacter baumannii, which is causing big problems right now in hospital settings, as well as gram-positive, such as MRSA, again, a big problem in hospital. You can also detect fastidious pathogens, things like chlamydia, select agents, as well as some viruses, especially those that cause gastroenteritis in humans and animals. The most important thing is the emerging microbes do not show any enhanced resistance to either antibiotics or biocides, which is what you would naturally use to kill those strains, things like bleach and alcohol. And they do not show any mutations that are due to growth in amoeba. Now, the system does have limitations. It will not work for anaerobic bacteria because we do need that top area to be aerated, and it will not likely to work for enveloped viruses that need a little bit of the membranes of mammalian cells. We are currently building a prototype device for testing in field deployment, and what it will have is, in addition to the 2 compartments, the top is going to have the swab attached to it, and a lower lock is going to allow the swab to sit in at the first notch where it will be separated from the bottom compartment with the amoeba using membrane. And once you take your sample, then you get into the second stage of the lock, and that allows the swab to break right through, and then you basically just mix it up by inverting up and down. And it's stable to sit there, then you can get it into the laboratory. The advantage of this system over what's available right now is, I mentioned, it can be kept at ambient temperature for any time duration you want. You're not limited by having to get it to the laboratory within 72 hours. It can also withstand temperature fluctuations, and there is no requirement for freezing or refrigeration or anything like that. In addition to that, bacterial pathogens maintain their clinical phenotype with no loss of virulence, and that's because they are growing intracellularly. And we have actually demonstrated that when they come out of this system, they have similar rates of survival in mammalian cells, just as typically grown macrophage strains. And macrophages are the body's natural immune cells, the first thing that will cause pathogens when they invade a human being. Now, since amoeba only feeds on bacteria, samples that are contaminated with patient or environmental flora are actually cleaned up during your transport, which is a bonus we were not thinking of when we developed this. And the system actually enriches for your pathogen of interest because it replicates very well within the amoeba. To summarize here, we're seeing this as a sample collection device from the field or hospital to the laboratory. And we see our customers here being either the military or civilian physicians that are deployed in areas that don't have access to highly refined microbiology laboratories. And again, the current practice is only typically uses a swab with a media. We also have an advantage here is the media that we have will work for all of those pathogens. We do not need a specific media for gram-positives or for gram-negatives. One media works for everything. In addition, because the amoeba will actually eat your environmental or medical flora, this system will decontaminate contaminated samples, and this is a huge, huge problem, especially in field locations where you can't get a clean sample. In addition, we see this also being used as a storage system in research laboratories because in addition to it not requiring freezing, it also will keep the samples, the pathogen samples, virulent. It will keep them in their natural clinical stage rather than having them become lab adapted. In addition to that, this system is just really very, very effective with every pathogen that we have put in it. And so there are many additional pathogens that will survive in there as well. Thank you very much for your time. My name is Sahar El-Etr, and if you need any additional information, please contact Catherine Elizondo at the IPO office in Lawrence Livermore National Lab.